Meathead arms or what?
There you go.
That's cheating. So do you guys know what the significance of a blue top tube is?
No.
Why we're using one of those?
Is that the ones? Oh no, it's not. So are those ones you have to fill up?
Okay.
Why are you shaking so bad there, Ed?
Oh,
So I was
because
looking at.
it's my
Here.
nerves in my neck.
Okay.
I shake like that all the time.
Good. Good news,
Actually, it's
maybe.
because he's not had the
So
meth
if you
lately.
look at this has sodium citrate in it. You guys know what's significant about sodium citrate?
Let's see.
They do. They just don't remember it because we mentioned it.
So sodium citrate is what they put in blood at the blood bank when you draw blood. And it prevents it from clotting. What it does is it binds to calcium. You guys are familiar with the clotting cascade, right? And calcium plays a crucial role in, I believe, the common pathway of the clotting cascade. So we have these vials have sodium citrate in them, keeps the blood from clotting, and that's so I can use it for something special tonight. Okay, so this is the microscope flex cam setup that I'll be using in lab today for the pathophysiology and anatomy physiology of pharmacology. This will be the first lecture of the paramedic students. We'll be looking at some histology of the nervous system. That's what we're talking about today, and we'll also be looking at red blood cells that you saw earlier. The blood was actually drawn from me, and I have actually taken this blood and citrated it as I talked about in the video. It's a citrated blood, and I'll be adding the blood to slides, and then what I will do is I will add various types of saline solution to that blood, and we get to see the effects on the red blood cells themselves. The blood will normally initially start out mixed in diluted. Actually, I'll dilute my blood in a 0.9% normal saline solution, and the red blood cells will look relatively normal. I then will move on and I'll add a 10% saline solution, 10% sodium chloride. That is a highly hypertonic, and I would expect fluid to shift out of the cells, and they would go through what's known as crenation. And then I'll go ahead and do another slide with blood and add distilled water to it, and I would expect lysis of the cells. And actually what will happen is the cell membranes will rupture, and there'll be kind of a round, ghostly, hard to appreciate image that will be left over of basically the cytoplasm within the cell, and we call those ghost cells after the red blood cell ruptures. So we should be able to appreciate those, and I'll also have, again, various histology slides of the nervous system and motor plates, parasympathetic and sympathetic ganglia. So the setup I have today is I've got a microscope here. You can see the microscope, and I have what's known as a flex camera here. I'll show you guys how that works. The flex camera is connected to the television here. You can see that through the auxiliary or AV in on the television, and as you can see I have an adapter for the flex cam placed on the microscope. So what I will do is I'll go ahead and take the flex camera here, if you guys can see. I'll take that flex camera, and the flex camera will then go ahead and be plugged in to the adapter like so, and that will put an image up on the television here. So let's go ahead and we'll go ahead and take out a slide. Let's see what we've got here. Got various slides, so we'll go ahead and pull out actually I've got a slide over here actually. Okay, and we will put nerve endings in motor plates into the microscope here. Trying to do this with one hand is kind of interesting. Okay, so now we're in. I'll just go ahead and point this up, and we will look around and find our tissue sample. And this is, if I remember correctly, this is actually a relatively small sample. Aha, there we go. Okay, so there we have This is actually skeletal muscle. And we'll go ahead and obviously the quality is going to be a little difficult on this iPad here, but there we go. It's fairly decent quality. So we have our muscle fibers. Don't really have a great amount of magnification quite yet. You can see all the muscle fibers, and we do have some neurons, and looks like maybe an end motor plate here. What I'm gonna go ahead and do is I'm gonna increase the magnification here. All right, and then I'm gonna go ahead and there we go. So now we're, I think, at about 40 power here. There we go. So we've got, looks to be a neuromuscular junction here, a little end motor plate here. Let's go ahead and look through all this. Aha, here we go. So I've got an end motor plate here, here, and here. So you're able to appreciate all that. And that's actually what I'll be doing for this entire lab today, is just kind of going through different slides, and we'll do the blood exercise. All right, guys, thanks for hanging in there. And here we just have a view of red blood cells in an isotonic solution, just kind of floating around. Thought I'd go ahead and show you guys what it kind of looks like.